just a little bit about me. So obviously, I'm being the employee of in Silico since actually April 2020. It's going to be 5 years. Prior to that, I was at other AI startups. So altogether, I've been in this space for a little over 12 years. And I'm also sort of interested in longevity as such as quite a few other of my colleagues at In Silico Medicine. So that's not something unique. Questions to be addressed in this presentation. Obviously, I want to sort of share a bit an update on the sort of major breakthrough and the drug discovery space. Want to talk about how was this achieved, what are the key success factors, and also how AI contributed, and obviously what will be the future of let's say pharmacological or biological intervention design with AI. I borrowed a very short, I would say, motto from our friend Sergey Young, from his book, which probably everyone already read. Pharma is indeed getting faster and smarter, and I will tell you more about how this is actually happening. Right, so this is the big breakthrough. I mean, obviously this was covered by most of the major media, more recently by economists as well, but just to very quickly summarize, the drug R&D is actually a very long, expensive, risky endeavor. And what In Silico Medicine is trying to do is actually drastically disrupt the way new drugs are developed. And this is essentially how we do it. So if you're not familiar with this story, this is the first AI-discovered, AI-designed molecule to ever enter phase 2 of clinical trials. It's not only first. I mean, by AI discovered, we mean that we identify a novel target, which was essentially implemented in fibrosis and was never explored in that context using some of our platforms. But we also designed the molecule from scratch using a generative platform called Chemistry 42. We have done this in less than 18 months. I mean, less than 18 months took us to the preclinical candidate nomination. Obviously, it took us another 12 months to complete the IND enabling studies to apply for phase 1. And we spent close to $5 million on actually doing this. And compared to how the pharma is normally doing, obviously pharma takes at least 4 to 5 years to reach a preclinical candidate phase. And the costs are probably close to 100 million per successful candidate. So as you can see, we have managed to sort of beat a record. And as it was announced recently by a CEO, nowadays we are able to nominate and develop preclinical candidate in close to 9 months. So we are already accelerating on this already very short timeline. And obviously, time is money in drug R&D. So if you can do things faster, you will also end up doing them much cheaper. Now, the question, how exactly did we do it? So we utilize our platforms. And I'm aware of the fact that there is quite a few boxes on the slide. And obviously, not every single platform was implemented in the story. But I can just highlight that the novel target was identified using PandaOmics platform. The molecule was designed with Chemistry 42 platform. And obviously, we had to run a number of tests using the other applications within the Chemistry 42, such as physics-based docking, admitox profiling, and a number of others. And I will come back to these tools later in my presentation. Previously, or the traditional pharma R&D, the way it was done was pretty much a hit and miss project. So there was large screens of huge libraries of molecules. Hits were identified, and then obviously teams of scientists would eventually slowly develop those molecules into what we have as drugs. So we have a slightly different approach. We obviously always are very focused on identifying specific targets, protein targets. Essentially, it could be receptors, enzymes, kinases, ion channels, a number of others. So we really want to understand on the fundamental level in biology, what exactly is the protein doing in the body? How exactly is it interacting with others? And what can be achieved if we successfully inhibit or activate this protein and what impact it will have on the body. So we are looking. So instead of looking for a needle in a haystack, we are actually designing the needle, if you like that metaphor. I also want to mention, and there was a very good presentation yesterday from Anton and Olivia, which went into depth of what exactly is AI and what are the different methods and the entire sort of evolutionary path. So I'm not going to spend too much time on the specific AI methods, but I just want to remind everyone that although many people nowadays use AI and LLMs interchangeably, the world of AI is actually much richer and much older than large language models. So when I will refer to AI methods, I will not specifically mean LLMs. We would not be able to do this without data, of course, and that's also the underlying sort of topic of this conference, because data is extremely important, especially if you have a data-driven approach as we have. So the data that sort of powered our drug R&D engine is actually summarized on this slide. So we have a large number of omics data. 98% of those are from public repositories. You might be familiar with some of them, like G or TCGA. We also generate our own proprietary omics data. So altogether, we have been able to put together 22,000 omics data sets. I think close to 80% of them manually curated. And obviously, we have a pretty big depository of chemistry data as well. So around 1 billion of structures that we have, somehow profiled or predicted, and then obviously more than 2 million compounds that were used to train our models with very specific and concrete in vitro and in vivo readouts. And we also process text. So we have been doing this ever since the company started in 2014. We still have a natural language processing team. So essentially, we are tracking a very large number of grant funding applications all over the globe, and PubMed and clinical trials and Wikipedia and many other sources. We do that mainly for that reason that there is already a pretty deep knowledge about much of the fundamental biology, which is already published. Sometimes the data is a bit scattered and hard to find, but having a good and reliable system is actually able to analyze this data, summarize it for us. And in silico has done more than 160 collaborations since 2014. These are just some of the examples. Obviously, major universities were always are very valuable partners. We have published more than 200 papers, which are to be found in peer-reviewed journals. But we are also very active in working with the industry and also technology companies on basically data processing, data analysis. So that always has been a very sort of important thing for us. Okay, so let's take this maybe step by step. So how do we identify targets? We use a systems biology tool called PandaOmics. What it actually is, it's a large depository of data, on top of which we have built more than 20. So it's actually in the current version 23 models, which help us define gene disease associations. Some of them are AI. Obviously, I will talk about that. Some of them are classical statistics bioinformatics. Around half of those are trained on omics data and use omics data as input. The other half is based on text. So essentially what we are doing, we're taking 20,000 protein coding genes and we try to match them to the indication of our interest. We can run the engine in reverse as well, which allows us to basically take a gene and score and rank 10,000 indications for the gene and provide multiple layers of evidence. In the end, we can filter those ranks and based on novelty, safety, druggability, accessibility, to ensure that the gene-protein hypotheses we generate are not only nice and clean, but they also potentially actionable as targets for drug discovery. This is a glance of some of the models that we have. Obviously, we are looking at, mutated submodules, which means that how are different mutations driving the expression in the cell or in the tissue. causal inference, we're looking at transcription factors. Gene expression obviously is a very important element in the analysis. And then we have developed and trained two deep neural networks that help us generate these gene disease associations. One of them is heterogeneous graph walk. It's a graph-based model, and a matrix factorization, which most of us use in the recommender systems in YouTube, Netflix, and so on. So we have been sort of creative in this space, and we were trying to not only use the methods that are already there and they are established, but we also always look to invent new methods that can help us solve these problems. So once we identify the target, and basically, maybe I can just add that some of the targets have already been validated to some extent in laboratories, which means we already understand how safe or how effective they are in a specific tissue. So once we do all that and we decide to invest into, let's say, what we call prosecuting of the target, we would use the generative chemistry platform called Chemistry 42. The first paper published on this topic actually dates back to 2016. And so just want to remind everyone that InSilico was doing generative AI way before it was cool. Actually, back in those days, it was actually not cool. And people actually thought this is an approach which will never work. And probably it would be the same until now if there would not be the GPT breakthrough, because obviously that helped us, I mean, the AI companies in general, a lot in terms of sort of evangelizing the technology. So essentially what we are doing is we have a target, and now we want to find a molecule or design a molecule that would be inhibiting or activating a specific target. So we use a combination of generative algorithms. Currently, we have 42 different generative algorithms. Some of them explore the chemical space. Some of them exploit the generated idea and try to basically navigate the chemical space into something which is safe, stable, druggable. But essentially what the user normally does is they imagine what an ideal molecule would look like in terms of physical chemical properties, and then instructs the system to generate ideas. Very often we generate millions of ideas, of course, and not every single idea is actually a good one. That's why we have built an entire series of reward functions, medicinal chemistry filters, and 2D and 3D descriptors, which means that in our context, the reward function is your virtual chemist that is actually trained to assess molecules that are generated and compare them to the original target product profile, which ensures that those ideas that do not satisfy the configuration would actually be filtered out and would not make it to the final selection. Obviously, there is a reinforcement learning element as well, which means the two different generative and reward functions talk. Different models generate different quality of molecules, and the reward function actually allocates more compute to those models to generate useful stuff, and actually take compute away from models which are not generating useful molecules in the context of the project. Basically, every single molecule is very closely scrutinized. I already mentioned that we have these medicinal chemistry filters, so we want to ensure that those molecules which make the short list, and very often we are talking about tens, maybe hundreds of molecules out of millions, are really drug-like and worth investing into synthesis and testing, and could potentially offer a good chemical starting place for the experiment. This is a more recent feature of the platform, but now we have, I think, close to 30 different parameters, which obviously describe what we call admitox properties. Admitox essentially means how is a specific molecule absorbed or distributed in the body, excreted from the body, how does it interact with the tissue, with other entities in a cell and an organism, and obviously the success or the failure of a specific drug candidate very much depends on how this molecule actually behaves in the body. So that's why we put a lot of emphasis on training these predictive models, which essentially allows us to understand the molecule even before we enter animal models or humans in a clinical trial. This essentially already has an impact on the industry. You may have read a recent a report from the group at the BCG, which essentially summarized a large number of AI-generated molecules and their success rate in phase 1, and the success rate of phase 1 was actually much higher than the average or the standard, over 80%. So, which means that AI does create interesting molecules which are safe by design, and obviously it's maybe a different story about efficacy, which is normally tested in phase 2, but at least we know that we can generate safe molecules. This is just again a glimpse at different machine learning methods that we use, either for the generative AI, but also for the reward function. Yesterday we had a good intro, so I will not spend too much time on those. So how the different platforms interact in an end-to-end engine. So obviously Pandomics is the system that generates the target hypothesis, and then chemistry 42 generates molecules. We do have to run this design, synthesize, test, and adjust the STA cycles, which means that even though AI already predicts quite a lot, and we know quite a lot about our molecules, we still need to undertake some of the wet lab experiments, synthesize physical molecules, test them in different assays, and then generate data which then can be fed back to the system and generate better quality molecules in the next iteration. But obviously, as you can see, the number of cycles and the months that are involved in those cycles is actually much lower than the industry standard. Allows us to test more hypotheses cheaply, and therefore design more molecules that could be potentially drugs. Now, one step forward is, and again, this was mentioned yesterday already, we also have a robotics lab, which is based in Szőllős, China. It is connected to the cloud, so essentially it's connected to all of our platforms. And what it essentially does is that every time we identify a novel target, which means a new protein never before explored in the context of a specific disease or a specific cell, we can very quickly set up experiments, some of them based on CRISPR, some of them knockouts, knockdowns, so different methods, and very quickly we can test the hypothesis in the actual living material, so different cell cultures. That essentially allows us to generate new data, which can be then fed back to Pandomics, to the cloud, and reanalyzed, and we can do all that in a closed loop pretty quickly. The next step for us is obviously to automate the synthesis for the small molecules, and hopefully at some point attach this ecosystem to, let's say, hospitals or some other places which actually take care of patients, so we can deliver this innovation to the patients directly. An update on another platform. So recently we also launched what we call generative biologics platform. The reason why we did it, obviously, very often you come across hypotheses and some proteins are just not druggable by small molecules. So we wanted to expand on the modality options, and therefore we sort of reuse some of the generative approaches we developed for small molecules and apply them to biomolecules. So right now we have a system which is able to generate alpha helical peptides, antibodies, and antibodies from scratch, or optimize existing molecules and optimize them for better properties such as binding affinity, developability, and so on. So we are greatly extending the modality options that we offer. I wanted to talk a little bit about Precious, because in the context of aging is obviously sort of the biggest project we have done to date. Yesterday we saw that in action, so Anton demonstrated what it actually does, so I'm not going to talk too much about this. Just wanted to remind that there is actually 3 models and 2 different publications which I recommend reading. The Precious 1 GPT was essentially developed to be an aging clock. Precious 2, sort of on top of that ability, also was capable of generating synthetic data or controls for some of the experiments where these controls were missing. And then the Precious 3 GPT already has also the small molecule emulation component integrated. Right. Wanted to cover a few case studies before we end. Not sure how much I'm doing on time, but just maybe 3 of them. So the first, very relevant in the context of aging, we have published this collaboration with University of Chicago, Liverpool, and also Morten's lab in Copenhagen, where we actually mapped 145 targets. So because obviously these are genes which we essentially identified as being implemented in age-related diseases, and then we mapped them to hallmarks of aging. Again, it's a very nice publication. I do encourage everyone to read this, if not already. So we managed to find, obviously, we have confirmed anti-aging properties of AKT1, mTOR, SIRT1, IGF-1, but we have also found a large number of other potential candidates. Then we published a study, so pretty much the entire research community can potentially explore some of these ideas. The other one is managed to identify novel targets for ALS. This was a very interesting study because here we collaborated with a couple of KOLs, I mean, global KOLs in the Johns Hopkins Mayo Clinic, also in Cambridge. But we also worked with the ANSA ALS, which is a patient group who were willing and very nice of them that they actually shared the data, which we then used for analysis, and we nominated, I think it was altogether 27 new targets potentially explorable for ALS. And there is an entire website dedicated to this project. So if you would like to see how a patient data can be effectively used to identify novel therapies or novel therapeutic options, then this is a very nice case study. And then one which made it to the news as well, where we obviously took AlphaFold. So obviously the idea was to see how powerful AlphaFold predictions are. So we identified together with the University of Toronto a novel target which we believed was strongly implemented in HCC, hepatocellular carcinoma. It was CDK20. There was no structure published about this target, so it was completely dark. We knew very little. So we used AlphaFold to predict the structure, and then we generated molecules based on that sort of homology model, essentially, of the protein. We have found, or we generated a number of nanomolar hits, so high potency hits, and we tested all of those in vitro validation in assays that were available at the University of Toronto, and we done that all in 30 days. Now, I guess I don't have to maybe explain like how actually quick this entire experiment was, because normally it could take a couple of years to basically get from a target to in vitro validation. So we have done it in 30 days just to demonstrate how powerful these approaches are. And I want to wrap up with a couple of maybe slides on the future. Obviously, we are not the only company that is in the space. We are probably the oldest company in the space, but not the only ones. So I'm sure you have seen BioAge, or making a big leap in actually AI position molecule for aging, and there is obviously the usual suspects such as Altos, Calico, but also smaller companies and in silico spin-out, Deep Longevity. I want to bring your attention to the ARDD Congress, which is probably the biggest at least in Europe, dedicated to aging research. And I think we already had 11 annual editions of this. This will be the 12th. Started small 12 years ago in Basel. Now it's really big, and I myself going to be there next year. So looking forward to meeting you. And we had a webinar, but it was already this week. So that's Yeah. We have a target identification course if you would like to see what we do, and it's obviously free. You can try that. It's about 8 hours of content, mostly videos explaining well, systems biology approach to therapeutic target discovery, and you also get access to some of our platforms which you can then use and explore in this context. Right. So thank you very much for your.
I have a quick question about the course which you mentioned. Is it okay for like beginners to take the course? Because you have very little to no idea.
No, it's supposed to walk you through.
Can I repeat the question? So, yeah.
Okay, so we have a question about the course itself. So it's actually a very sort of, I would say, easy to comprehend approach to target discovery. So essentially, it takes you through each step, explains why and how each step is sort of executed. And yeah, I mean, you know, it's pretty much for everyone. We have high school students doing this and enjoying it.
There's a question from George on
Yeah. Yes, George, you can ask your question.
Yes, thanks for this really exciting talk. I'm wondering, you are doing lots of predictions, regression-based, classification-based. How do you deal with uncertainty? Did you, for example, look into conformal predictors? That's a pretty recent framework, or maybe Gaussian approaches.
Okay, thanks, Lord. I don't have every single technical detail about these predictions, but obviously we are looking at a number of conformations that we are trying to predict with different models. But if you would be interested, we can continue this discussion offline. I can connect you with the team which is developing these models. And you know, also watching your talk yesterday, I think there is a lot of overlap of what we do also in terms of bioinformatics. Especially intervention impact prediction. So we'll be happy to chat after the conference as well.
Yeah, very good. I would get in contact. I've also been at the AARD and talked to Alex and so on. So I think it would be nice to have a meeting, maybe even before December or before Christmas or after. Thank you.
Yeah, thank you.
Okay, thank you, Jan. It's better that you come here. You can speak there. Yeah, normally it works.
Yeah, thanks for the very impressive talk. I actually remember also one of the very first ARD conferences. You had like these backpacks with these logos, and actually people still today asking me the train, oh, what does this mean? So maybe very let's say more popular question. So if someone asks me, so but what is actually what's the outcome? So could you maybe could you name a drug which has been, or treatment that has been, well, which is thanks to you, or which you have facilitated come into action?
Okay, so I wish I could tell you an approved drug that was already discovered by us, but that's not the case, of course, because although we can speed up and automate much of the preclinical work and discovery work, the clinical trials are still highly regulated endeavor. So therefore, it still does take quite a few years to actually get a drug properly tested and approved. So we are not there yet. And to our knowledge, none of the AI drug discovery companies have managed to reach the market. But we are already in phase 2. We already had a positive readout meeting both of the primary endpoints. So if we are continuing to be successful as this, we probably could have a product approved in 2 years. And I think we might be the first ones to actually reach this stage.